Gerhard is a theoretical physicist. In 2001, he published an influential Nature letter entitled "Water Conduction Through the Hydrophobic Channel of a Carbon Nanotube" that was cited over 3,300 times as of this morning. This paper exemplifies the strategy in many of his studies, which start with hard biophysical measurements such as mass, dimensions, and chemical properties, but he often then pushes the system to see how far it might respond in a real-world context, informing their later application. No current example is more relevant than the structure of the SARS-CoV-2 spike protein that hopefully everyone in this call has already been vaccinated against. Gerhard's recent work begins with the image-based measurements of cryo-EM coordinates and extends to real-world motion through simulations within known biophysical constraints. One of my advisors said that the future of biology will be revealed through the lens of Pixar-like animators who can help us biologists to dream about how systems actually work. And I really see this vision beautifully realized in Gerhard's work. Gerhard, please go ahead.
Well, thank you, Dan. This was very kind of you, first for this introduction, but also for the opportunity to present my work to an audience that I would not normally have a chance to talk to and interact with. So I'm very excited to be part of this group, and I really look forward to the discussions and any future feedback. So as Dan already said, I have a bit of an unusual background for this type of workshop, but actually we have a very strong interest in many of the techniques that have come out of this community, and there have also a strong interest in the kinds of applications that you are pursuing, and I hope that will become clear through my talk. So in the last several years, we have witnessed really a number of amazing developments, and in particular in the area of electron microscopy, and there also very strongly in the area of cryo electron tomography. Electron tomography is a technique that allows us to resolve structures in cells down to the molecular level, and on the left you see just a brief reminder of the basic approach. This is a tomographic approach. The same sample is imaged from multiple orientations, and then rather highly developed computational techniques, a three-dimensional density is reconstructed. But the primary workhorse is a form of back projection. And with this tomographic reconstruction, and then subsequent averaging of resolutions down to the near atomic level can now be achieved, certainly in the sub-nanometer to a few angstrom level of resolutions. And that has made it possible to resolve structures such as this nuclear pore complex, very recently coming out of my colleagues' lab here at the Max Planck Institute, Martin Beck and Allegretti et al. And so what we do, however, has also undergone quite dramatic developments in the last few years. And this is, we use computer modeling and computer simulations. And I hope you can see this animation that I'm showing you of some recent work that we have been doing. This is a computer model, molecular dynamics simulation of a dense protein solution at roughly the density of the cytosol, so at about 150 milligrams per milliliter. And we can simulate this at the level of fully atomistic representation. So this includes all the water molecules, all the ions, and several hundreds of proteins, which ends up in a simulated volume of several millions of atoms. And we can then, as you can see, maybe I just run this again. So let's see if this works. Yeah, you can then see the proteins interact with each other, form transient complexes, and then diffuse through the system. So since I think it's fair to assume that most of you are not familiar with the technique of these kinds of molecular dynamic simulations, I want to give you just a very brief summary of what this is, because it is actually quite simple, at least at the core. So we represent molecules as a collection of atoms, and we describe these atoms by their positions in space as a function of time. These interactions interact with each other according to a potential energy function, and this energy functions includes terms for chemical bonds. This would be harmonic springs. We have charges for the electrostatics, and other terms that describe interactions of atoms with each other, both bonded and non-bonded. And then from these potential functions, we get the force by taking the gradient, and then we simply solve Newton's equations of motion. Mass times acceleration equals force, and we solve them for, as I will show, for about a billion time steps or so. So very long time integration, and those give us trajectories of the atoms and the molecules through space and time, from which we then calculate the properties of interest: structural properties, thermodynamics, dynamic properties, rates, and spectral properties. And to give you a bit of an idea, here is a movie of a simulation done by Judith Warnau, where she looks at one of the core processes in our cells, which is in oxphos in respiration, the transfer of electrons, and here from reducing agent to the quinone, and then the removal of the quinol from a complex one. And these types of processes can be simulated and studied. And what I would argue for that these types of simulations, because they rely on a physical model of physical reality, can be considered a computer experiment, and that led one of the pioneers in molecular simulations, Klaus Schulten, late Klaus Schulten, to refer to them as computational microscopes. So in the following 20 minutes or so, we will be looking together through a computational microscope, but we will not be completely unaided by experiment, because these computational microscopes, as I will argue, team up very nicely with electron microscopes, and increasingly also with super resolution optical microscopy. And whereas electron microscopy usually gives us a static view of flash frozen samples, the molecular dynamic simulations add dynamics and additional detail to these static representations. And this, of course, at some level has quite a history, and in particular in studies of individual proteins and their functions. For instance, here a work collaboration with the labs of Arne Müller and Robert Tampe on the function of an ABC transporter is involved in multi-drug export, where structural data from single particle electron microscopy were integrated into dynamic models and then related to the processing function of this machine. But in the following, I will show you that we can also very nicely integrate lower resolution tomographic data into our computational models, and in that way arrive at a hopefully more detailed and richer picture of complex systems. And the ones that I will show to you are cell-cell junctions, in particular desmosomes as a very strong type of cell-cell junction, their structure dynamics and their mechanics. And then, as Dan already alluded to, our work on SARS-CoV-2 spike and its interactions with the host receptor and antibodies. So let's start out with the structure and dynamics of cell-cell junctions. Desmosomes are among the strongest cell-cell junctions, and they are found in tissues that are subject to mechanical stress, such as heart, but also epithelia. And they're formed by two classes of caterins, the so-called desmosomal caterins, desmocolin and desmoglein. And on the bottom left, you can see a tomographic image from the Frangakis lab, our collaborators here in Frankfurt, of a desmosome in mouse liver. And we have the two plasma membranes at the top and at the bottom. There is a very dense midline, and then through in this hand segmented, hand-colored image, you can see these somewhat curved structures, and these are desmocolins and desmogleins. If one then does a subtomogram averaging of these structures, then one sees there is a form of regularity in them. They have a near square like a lattice structure. However, the regularity, as the image on the left suggests, does not extend very far. There is enough irregularity in them that a subtomogram averaging with a reasonable number of subtomograms does not lead to high resolution structures. Indeed, the resolution was limited to over the 2 nanometers, so really quite coarse. And that meant that it's rather difficult to obtain a detailed structural view of the organization of the caterins in this cell-cell junction. Matt Sikora built caterin structures into them and obtained a reasonable representation also of the subtomogram density, but he quickly realized that there was not enough information for a complete representation. Still, these W, S, and Y shapes that have been reported previously and have been seen also in the Frangakis tomograms emerged quite naturally from the different models. But as I said, there is enough ambiguity in the modeling that it is not clear whether we actually have the right model. So Matt then went back and considered all possible, as many possible arrangements as he could come up with that were roughly consistent with the kinds of subtomogram average structures that we had obtained from electron tomography. And you can see the kinds of arrangements of these interactions in the dense midplane. And we have different orientations of the interactions because of this hockey stick shape of the individual caterins. The most important ones are, and because I will refer to them also later, are the parallel ones where the hockey sticks all point in the same direction, and the anti-parallel ones where you have lines of sticks that run in opposite direction in the midplane. And you can see in 3 dimensions that has a quite profound influence. In one case, you have a nicely orientated arrangement, and the other an anti-parallel cross-shaped arrangement. And then Matt looked at consistency with all kinds of data, both structural and biochemical, and ranked them. And even that was not sufficient to rule out all possibilities. Instead, there was 2 classes that remained. And so for that, he then went to atomistic simulations to get a more detailed view of what is possible and what these structures might actually look like. So here I'm trying to give you an idea of how these simulation systems are set up. So we start out with a single caterin dimer we replicate it according to a rule, okay, in a form of a 2D crystalline arrangement initially, and then we put these structures into a simulation box. We add the water and iron, and then we periodically replicate. This is like a unit cell. We always have periodic boundary conditions, and that leads to a lattice-like structure of coherence, and then we solve Newton's equations of motions of the entire system, including the water and the ions and everything, and then look at the dynamics. And so this is repeated on this slide, and again, I hope this transmits. So this is nice and interesting, and you can immediately see the dense midline here. So that is a nice feature that emerges directly. This is the place where we have the strongest interactions. But what we could also see is that these two arrangements, parallel and anti-parallel, had very different properties in terms of structure and in terms of their mechanics. And the parallel arrangement preferred rather tight 24 nanometer distances between the two plasma membranes, whereas the anti-parallel arrangement matched exactly the experimentally measured membrane spacing of 32 nanometers. Interestingly, adherence junctions, which are mechanically less strong, are at about 25 nanometers distance, so close to the parallel arrangement. This is reflected in the mechanical stiffness. We can look at the force response, and we see that the anti-parallel arrangement is stronger in both vertical as well as in horizontal shearing type motions. And this is illustrated in a bit more detail on this slide, how these structures respond to force when you this is vertical force, so compressive and expansive force. And as you can see, the parallel arrangement on the top line responds rather smoothly. It poses very limited resistance to the application of force. By contrast, the bottom structures are for the anti-parallel system under compression and expansion, and it resists both quite strongly, and it does so by tightening up under compression and expanding straight under vertical pull. And this now shows that the truss-like architecture of the desmosome, which is consistent with the tomographic view, this brings us back to the starting point, appears to be the main factor giving these cell-cell junctions their high mechanical stability. So now I'm going to shift gears and move on to our SARS-CoV-2 studies. And I think at this point in time, I probably don't have to explain very much to you how SARS enters cells. The important point here is that the spike protein is a main player in both the recognition of the cell surface receptors and as the fusion protein that then mediates the entry into the cell either through an endocytic pathway or through direct fusion with the plasma membrane. So very soon after the initial reports of SARS came out, there were already the first structures coming out, also high resolution cryo-EM structures of the ectodomain of the spike protein, very important, of course, for the vaccine development. And so we use those also in the very early phase as a starting point. However, there were important parts of the protein missing, and these were the connections of this large domain to the viral envelope. And this stalk-like structure we modeled as a coiled coil. However, the bioinformatics analysis identified several interruptions of these coil structures, and those were modeled accordingly. And we also had to model the transmembrane anchor, and very importantly, we had to add the post-translational modifications, which include an extensive cover of glycans as well as palmitoylation. This initial model, partly based on high resolution experiments, partly based on real bioinformatics analysis, was then the starting point for molecular dynamics simulations. And I don't go into the technical details, but here you see our simulation system. It's a hexagonal unit cell with 4 spike proteins in an irregular arrangement, interacting both with the membrane, so anchored in the membrane, and interacting also with each other, so quite densely packed. And what we saw in these simulations was that spike was actually remarkably flexible in its connection to the cell membrane, and it exhibited several very clear joints where flexibility was initiated. And this is illustrated here in these rigid body superpositions of different parts of the protein. So on the left, you see a superposition of the top large domain, and then you can see the fraying of this stalk-like structure. Here you see a superposition of the top quark structure, and again fraying and so on. And you can see there is flexibility, very high flexibility actually, throughout the protein, because these joints really have quite a wide range in angle. What happened, this is probably typical of pandemic science, is that in the same building, unbeknownst to us, our colleagues were already working on tomographic imaging of live virus collected from infected cells, and they obtained quite beautiful pictures. And when we finally found out that we were effectively working on the same system, but the different points of view, we started to compare notes. And what we found was that there was really remarkable consistency between the structures seen at stunning resolution directly from the tomograms and the structures that we saw in our molecular modeling of the spike protein. And as I pointed out, there's this high degree of flexibility, and it results in quite significant and very clearly visible bending of the protein on the surface of the virus, as seen in the Cryo EM tomograms. Indeed, we could take our molecular dynamic simulation structures and place them directly without any further bending or adjustment into the densities obtained by 3D reconstruction from electron tomography. As a first remarkable level of resolution achieved electron microscopically, and then really this idea of the computational microscope showing that we can achieve a high degree of consistency. So our explanation of this is the high degree of flexibility is the likeliest explanation is that it adds possibilities for the virus to interact with multiple receptors in an irregular arrangement. So effectively, it enhances the avidity of the virus host interactions. And we've seen another indication of that. Virus spike proteins can actually cluster at the surface of virions, which again would enhance the avidity. Another interesting point that one can see in these tomographic views, the tomographic densities, is that there are these very significant protrusions at the surface of the spike protein. And of course, now with these computational models at hand, which are fully glycosylated, we can check whether these protrusions are indeed the glycan code of the protein. And that turned out to be the case. We had a remarkable level of consistency between the glycans glycosylation sites inferred from mass spectrometry and then modeled into our computational model with the 3D densities, electro-optical densities from the tomography. So we can now be quite sure that we have full glycosylation, including at the stalk, which was not so certain when we started out. The effect of this glycosylation is quite profound. It is illustrated in this view here at different levels of contouring. Sorry. So you see the spike at the surface of the protein in different blue tones, and then in green you see the glycans. And so we contoured the density in 3 dimensions according to the occupancy of the different voxels. At the high contour level, you can see a significant fraction of the protein surface exposed. At intermediate to low contour levels, almost the entire protein surface disappears behind this curtain of glycans, which means dynamically they really cover almost the entire surface, except those parts that are well known, like the receptor binding domain. That is, of course, a primary at the top as a primary interaction site with the ACE2 receptor. So that just a little detour now was the basis for another, I think, for us at least, a very exciting work. It's pushed us into a new direction. That is to ask the question, what are good epitopes for antibodies that at the surface of the spike protein? And we, to answer this question, we looked at not only the accessibility, but also elements of how flexible these structures are, how conserved the sequence is, and how typical these sequences are, whether they carry the signature of antibody interactions. And we turned this into an epitope score. And what we found was that when we published this, a very significant fraction of all the predicted epitopes were already captured by known antibodies. By now, I think nearly all of them have been reported as antibody binding sites, and this is illustrated here. So the last scientific point that I would like to make is that this glycosylation can have also have dual roles, because not only the spike protein is glycosylated, also the human cell surface proteins tend to be glycosylated, including the ACE2 receptor. And what we found was by looking at the effect of ACE2 glycosylation on its interactions with the spike protein, that some of these glycans, in particular the one at the N90 site, actually have a certain level of protective function against spike binding. And indeed, we then learned, going to the literature, that mutations of this site that remove the glycan are actually associated with higher susceptibility. So with that, I hope I can give you at least an idea of how we can go from tomographic views, modeling, to address questions of some also practical relevance in the broader field. And with that, I would like to wrap up. I started out by stating that molecular dynamics simulations and cryo electron tomography complement each other. I hope I have convinced you that this is indeed the case. I showed you that we could build a model of a desmosome and show that the truss-like cadherin architecture gives them a very high level of mechanical stability. I also showed you some work on SARS-CoV-2 spike and the connection of our work with tomographic studies. And I hope that this type of very systematic approach also sets a basis for further studies for instance, antibody and vaccine development. So with that, I want to thank the people who did the work. I already mentioned Matt Sikora. He did not only the desmosome simulations, but he was the lead person on the simulation side for the spike protein as well. Reza Mektipur did the simulations of AC2 with spike interaction, as well as the ABC transporter that I showed very briefly. The EM work was carried out for the desmosome by Achilleas Rangakis, and for the spike proteins, this was really a fantastic collaboration with Martin Beck and his group, and in particular with Beata Toronova, as well as with the group of Jacomine Kreinze-Locker at the Paul-Ehrlich Institute. And so with that, I will stop here. Maybe I should say thank you also to the other members of my group, really in this spike and these simulations came in with full force, and that's Sören von Bülow, Florian Blanc, Michael Gecht, and Roberto Cudino. Here I stop and take your questions. Thank you.
Thanks, Gerhard. That was wonderful. What a great kickoff to this meeting. I think it illustrates the importance of cross-functional collaboration, working with microscopists, biologists, theoretical physicists to kind of connect the dots. So wonderful. I was glad that the videos played well as well, because that's always a risk with these sort of fantastic presentations that they get stuck somewhere. So maybe I can start with one question about the spike protein component of the presentation. You made the argument that flexibility is an asset to the spike protein with respect to substrate binding, and I haven't heard much about that as a strategy for antibody therapeutics. So you did mention that most of the predicted epitopes have been experimentally kind of confirmed as true antibody binding sites. But is there a strategy to, for example, strengthen or increase the rigidity of the spike protein as a antiviral strategy? So targeting that using that as an immunogen to kind of create antibodies that may, for example, stabilize that structure.
Yeah, so this is obviously something that should be pursued, and I believe, I mean, in a way, it has already been pursued, because one of the key differences between some of the vaccines and others is that they use modified versions, and in particular, the mRNA versions of the vaccine use stabilized spike. So there is, and these stabilizing mutations were introduced for structural studies. So they have been screened and sometimes also rationally introduced to stabilize the protein. So it turns out, I mean, by the success of that strategy, so mRNA vaccines by and large, from the first generation produced a very good immune response. Stabilization would appear to be a good strategy, and that's primarily stabilization with respect to these large-scale conformational changes that the spike protein tends to undergo. This is the partial opening up. So there's all kinds of dynamics that I did not discuss here. But I think now to your broader question, my collaborators and my view is that indeed this is a strategy that should be pursued also more systematically now that maybe more resources for these kinds of studies are becoming available, because I mean, we know from other viruses such as HIV, of course, is a particularly terrible case, that they use the opposite strategy to present highly dynamic and sequence variable elements to attract the attention of the immune system, that then of course responds to those elements is utterly useless. Whereas the structured parts and the more functional parts are not normally targeted. So I think that to revert that and use engineered systems with presenting the epitopes in a still natural form, but maybe with less dynamics, I think is a very good suggestion.
Thanks. Are there comments or questions from the audience? I'm not sure. This is of course, I'm the first one to do this, so we're sort of learning how to run this meeting with comments or questions. But if somebody wants to type something into the chat menu or so, that's also fine.
Yeah, exactly. They can either directly unmute and ask, or just type the question in the chat window, or raise hand.
Oh, I noticed there's also in the participant list, there's a raise hand function. That's probably like a most effective way if there are other questions.
Yeah, let me monitor that one.
Maybe I can go back if there's no action in the audience. Just one more question around the desmosome, the earlier studies that you described. So as you kind of extrapolate and extrapolate and extrapolate, you get to of course a point that's further from the original measurements that are made around the structure of the desmosome components, and of course you kind of get into the Wild West where you have maybe less confidence in reality. So at some point, you must be able to do functional tests that go beyond imaging or structural analysis that can help you to kind of build confidence in the matrix that you created, the kind of heat map of confidence. And so how do you approach that sort of confidence building when you kind of back translate to reality?
Yeah, so this is of course a very critical element, and I mean the standard approach is to then go back and look at strong interactions and do mutations, and then look at the functional consequences of those in the context of the organism, for instance. So if we as you could see, we had these higher order interactions, and one would then investigate each of these interactions and see if we can either strengthen them or weaken them even more, and then use those proposed mutations for tests. But the problem, of course, is that in the context of such a system, this is very difficult because, as we learned, and I think the field has accepted and recognized a long time ago, each individual interaction is actually really very weak. Like, these are not your nanomolar protein binders, but these pairwise interactions are individually very transient. It's just, again, a form of avidity. It's the sum of them. And so that makes also that approach really rather challenging. So that means proper validation is far from trivial for these very dynamic structures, because they do not respond to mutational changes in the way that a nanomolar complex would respond by, if you introduce some like charges on the two faces, and they falls apart. And this is just not the case. It may just move. So the other way of doing it, of course, is to add labels, for instance, and this can be fluorescence labels. So this is where maybe this community might also come in, or spin labels to resolve individual elements of the structure. So you can do some form of distance measurements like that. But these are all rather challenging
Perfect. Thanks so much, Gerhard. Before the meeting started, a few of us were talking about challenges, and there's still many open challenges in segmentation in this field. So don't consider the the applications that Gerhard's talking about as solved problems from the segmentation component. So do reach out if this is an exciting challenge for you. And in the consideration of time, I'll just close this session. Thanks so much, Gerhard. Wonderful start to the meeting, and thanks for calling in from Europe and getting this meeting off to a great start. Take care.
Okay.
Bye-bye.
Okay, so good afternoon. This is Dimitri Metaxas. I'm the other co-organizer here. So I'm very happy to introduce Jens Richter, who is a professor of engineering at engineering science at the University of Oxford. And with his appointment held jointly between the Institute of Biomedical Engineering and the Nuffield Department of Medicine. He's a group leader at the Big Data Institute and affiliated to the Ludwig Institute of Cancer Research and the Wellcome Center as an adjunct member. Previously, he was senior scientist, research science manager at GE Global Research, where I met him like over 20 years ago. His research interests lie in enabling biomedical imaging through the development of new algorithms and novel computational platforms. And his current focus is to improve mechanistic understanding of cancer and patient care through quantitative analysis of image data, which is a great goal, of course. And he's the co-director of the Oxford EPA-SRC Center for Doctoral Training in Health Data Science, and visited him like a few years ago, and that's a great environment to be. So Jens is going to talk to us about a very exciting topic, of course, that you all are aware about. This is AI and how it opens new horizon for histopathology. So Jens, thanks for giving us a talk.
Well, thank you so much for the introduction. Yeah. I can hope you can see my slides.
Yeah.
Yes.
Yes. So maybe you need to make it bigger because we see your screen and the.
Oh gosh.